So, welcome back to the deep dive. Today we're really going off the map. I mean, if you open a standard microbiology textbook, you have your bacteria, your viruses, and your well-behaved fungi.
Right, the ones that follow the rules.
Exactly. The yeasts, the molds, the dermatophytes, they fit into neat little taxonomic boxes. But then you have chapter 66 of Murray's Medical Microbiology.
Yes, the island of misfit toys. That is, honestly, the best way to describe it.
It really is.
Yeah.
We're looking at this wild collection of organisms that are usually grouped with fungi in medical education. And they're treated by medical mycology departments, but biologically, I mean, they are a complete mess.
A total mess. Some of them are algae, some are, parasites that we've just conveniently decided to call fungi, and some are these genetic outliers that completely defy the standard definition of life kingdoms.
And so our mission for this deep dive is to essentially sort through all this chaos. We're anchoring this on Murray chapter 66, which is titled Fungal and Fungal-Like Infections of Unusual or Uncertain Etiology.
And we are also cross-referencing with jirovecii, specifically chapter 45, to really flesh out the clinical picture and the basic mycology definitions.
Right. And for you listening, whether you are a medical student prepping for boards or a clinician out in the field, you really do not want to gloss over this chapter. I mean, these organisms are the ultimate gotcha questions.
Oh, absolutely. They're those classic distractors, the ones that look like one thing. Maybe they look exactly like a standard fungus on a slide, but they require a completely different treatment approach.
Because if you treat, say, a pythium infection, like a standard fungal infection,
your patient loses their leg. It really is that high stakes.
Yeah. So we're going to categorize these misfits for you to make them memorable. We have the great pretender, the self-limited giant, the algae, the dolphin disease, and the genuinely terrifying water molds.
It sounds like a lineup of comic book villains.
It kind of does. Let's start with the biggest plot twist in the history of medical mycology, the great pretender, the organism formerly known as a protozoan.
Right, Pneumocystis jirovecii.
I remember looking at older textbooks, and I mean some really old ones I found in the library, where Pneumocystis was actually listed right next to toxoplasma and malaria.
Yeah.
It was considered a parasite.
And, for good reason at the time, morphologically, under the microscope, it behaves like a protozoan. It has a life cycle with these delicate trophozoates and these thicker-walled cysts.
And it responds to antiprotozoal drugs, which just reinforced that idea.
Exactly. But then the genomic era arrived. We started actually sequencing the ribosomal RNA, and the entire tree of life got rearranged.
The DNA doesn't lie.
No, it doesn't. And it turns out Pneumocystis shares far more DNA homology with the Ascomycete, the sac fungi, than it does with any parasite.
So it is officially, undeniably a fungus.
Officially a fungus. But it's a fungus that absolutely refuses to act like one. And this brings us to what is probably the single most important pharmacological concept for this entire deep dive.
Yes, the cell membrane.
Right.
Yeah.
If you take away just one thing from this section, let it be this. Pneumocystis does not have ergosterol in its cell membrane.
Which is wild, because ergosterol is the defining lipid of the fungal cell membrane. It's the target for almost everything we use.
Precisely. Think about how our heavy hitter amphotericin B works. It literally binds to ergosterol to punch physical holes in the cell membrane.
And the azoles, like fluconazole or itraconazole, they work by inhibiting the synthesis of ergosterol.
But Pneumocystis doesn't make ergosterol. It actually scavenges cholesterol from the human host and incorporates that into its membrane instead.
So if you give a patient with Pneumocystis pneumonia, or PCP, if you give them fluconazole or amphotericin B,
you are doing absolutely nothing to the bug. It's like shooting water at a brick wall. The drug has no target.
So to treat this fungus, we have to use antibiotics, specifically trimethoprim-sulfamethoxazole or TMP-SMX.
Which is a drug that targets the folate synthesis pathway. It's a very bacterial or protozoal target.
It's a fungus treated with a classical antibacterial. That paradox right there is bread and butter for exam writers. You will see that question.
Guaranteed. Now, let's help you visualize this organism, because diagnosing it is another weird challenge. You cannot culture it. You can't just swipe a patient sample onto a sabrewed agar plate and wait for nice, fuzzy colonies to grow.
No, because it's an obligate human pathogen. It needs a living human lung to survive. No one has found an environmental reservoir yet.
Right. So to diagnose it, you have to see it directly in tissue or fluid. And there are two main microscopic forms you're looking for. You have the trophic forms, or trophozoites.
Which are these very flimsy, thin-walled, amoeboid shapes that are actually really hard to see on standard stains.
They are. And then you have the cysts. The cysts are the money shot for diagnosis.
Let's talk about those cysts. They are about 5 microns in diameter. And the classic description, the one that should immediately trigger a mental alarm bell when you read it in a question stem, is the crushed ping pong ball.
I love that visual anchor. Paint that picture for us. Why ping pong balls?
So typically you're using a silver stain for this, like the Gamorre methenamine silver, or GMS stain. The silver binds to the carbohydrates in the cyst wall, turning it stark black against a green background.
But because these cysts in the lung are often empty or they've collapsed in on themselves, they aren't perfect spheres.
Exactly. They look like black circles that have been indented or crushed. They look like little cups or crushed ping pong balls.
And if you use a different stain, let's say a Giemsa stain or toluidine blue,
then you actually see what's inside the cyst. You see the intracystic bodies. There are usually up to 8 of them in a fully mature cyst. It looks kind of like a tiny little bag of marbles.
Okay, so we have these crushed ping pong balls in the lung, but it's important to note they aren't invading the tissue. They aren't eating through the lung parenchyma the way a mold like Aspergillus might.
No, and that's the key distinction for the pathology. Pneumocystis is almost entirely extracellular. It just hangs out in the alveolar spaces.
Specifically in the surfactant layer, it attaches itself tightly to the type 1 pneumocytes, the cells that line the alveoli. The actual tissue damage comes mostly from the body's own immune reaction to it and from the physical blockage it creates.
It's essentially suffocation by slime.
Crude, but highly accurate. In an immunocompromised patient, and we're specifically talking about HIV patients when their CD4 count drops below 200, or patients on heavy immunosuppressive drugs, the organism just proliferates unchecked.
Because the cellular immunity is gone. The macrophages can't clear it.
Right. So the alveoli fills up with this thick, foamy, honeycomb-like exudate. It's this nasty mix of the replicating organisms, dead immune cells, and cast-off surfactant debris.
And that completely blocks gas exchange across the alveolar membrane.
Which leads to a very classic clinical mismatch. You have a patient who is severely hypoxic. They are blue, they are gasping for air, their oxygen saturation is tanking.
But when you put your stethoscope on their chest to listen to their lungs,
it sounds surprisingly clear. You might hear some faint crackles, but there's no heavy consolidation or massive wheezing that matches the absolute severity of their hypoxia.
And then you get the chest x-ray, and you see that classic bilateral interstitial infiltrate, the ground glass opacity.
Ground glass opacity is just a radiologist's fancy way of saying a hazy, diffuse whiteness spreading through the lungs.
So just to recap the great pretender for you, it is a fungus, but it uses cholesterol instead of ergosterol.
So azoles and amphotericin won't work.
It looks like crushed ping pong balls on a silver stain. It causes a foamy alveolar block in patients with severe immunosuppression, particularly a CD4 count under 200. And you treat it with TMP-SMX.
Spot on. And just remember, because it's deep down in the lung alveoli, a standard throat swab won't cut it for diagnosis. You need to get down there. You need a bronchoalveolar lavage, a BAL, or at least an induced sputum sample.
All right, let's move from the most common misfit we see clinically to perhaps the strangest biological phenomena in the entire chapter. Segment 2, mycoses.
This one is fascinating. This is the self-limited giant. The organism is Mycoses crescens, which in a lot of older literature is referred to as Amansia.
So if you see Amansia in a question bank, it's the same thing.
Exactly. This is a fungus that normally lives as a saprophyte in the soil. It's usually a problem for rodents, ground squirrels, beavers, mice. It's a zoonotic reservoir.
But humans can accidentally get it by inhaling the dust from that soil. And what happens next is just biological madness. Because usually when a fungal spore gets into the body, it replicates. It buds to make more yeast, or it germinates to make hyphae, and it spreads.
Right. That's what a pathogen does. But Mycoses does none of that. You inhale a tiny little conidium, maybe 2 to 4 microns across. Very small.
Easily reaches the deep alveoli.
Yes. And once it hits the warm 37 degrees Celsius temperature of the human lung, it completely stops replicating, doesn't divide, it swells.
It just puffs up.
It transforms into this structure called an adiaconidium, and it grows from that initial 2 microns all the way up to 200 or even 400 microns in diameter.
Which is absurd. Just to give some scale for you listening, a normal red blood cell is about 7 microns wide. So we are talking about a single inhaled spore swelling to the size of a grain of sand. You can literally see a single cell with the naked eye.
It is titanic on a microscopic scale. And as it swells to this massive size, the cell wall gets incredibly thick. We're talking a wall that is 20 to 70 microns thick all by itself. I always call this one the balloon that never pops.
It just expands and sits there in the lung tissue.
And because it fundamentally doesn't replicate at that temperature, it doesn't infest the lung. It doesn't spread to other organs. It's usually just one or maybe a few isolated giant spheres.
So the human immune system recognizes this massive foreign object and walls it off. It forms a granuloma around it.
Exactly. And clinically, this usually presents as an incidental finding. It's a classic coin lesion. You have a patient who gets a routine chest x-ray, maybe for life insurance or pre-op clearance. The doctor sees a distinct round spot on the lung and immediately thinks, oh no, lung cancer.
Or maybe a tuberculoma.
Right. So they send the patient for a biopsy or a wedge resection to cut it out. And the pathologist looks at it under the microscope, and instead of a tumor, they find this massive, impossibly thick-walled empty sphere.
The empty part is the crucial differentiator here, isn't it?
It is the ultimate exam key. There are other fungi and organisms that make large spheres in human tissue. Coccidioids makes a spherule. Rhinosporidium, which we'll cover in a bit, makes a sporangium. But both of those structures are full of endospores.
Right, they're pregnant with hundreds or thousands of baby spores waiting to burst out.
But Atypical is totally empty inside. It has no endospores. It is just a sterile, giant, empty shell causing a localized foreign body reaction.
And so what is the treatment once you find it?
Absolutely nothing. It's entirely self-limited. The fact that you just excised it for the biopsy means you've cured the patient. It can't replicate, so it can't come back. For the fungus, inhaling into a human is a complete biological dead end.
I love that. It's terrifyingly large, but practically harmless in most cases. Okay, let's shift gears completely. We are leaving the lungs, and we're heading to the water. Or maybe the garden. Segment 3. The algal infections.
This is the section that always confuses students. I mean, why are we studying algae in a medical microbiology course? Usually, algae are a topic for marine biologists or botanists.
Right, they're plant-like. They belong in ponds.
But there are two specific genera of algae that have actually crossed the line into human pathology, chlorella and protozoa.
And the absolute easiest way to keep these two apart in your head is the color palette. One of them stays true to its algal roots, and the other has sort of gone over to the dark side.
That's a great conceptual anchor. Let's start with the true alga, chlorellosis, caused by the chlorella species. This organism still has its chloroplasts. It is still fully capable of photosynthesis.
Which leads to probably the wildest clinical sign in this entire textbook chapter. If a patient has a chlorella infection in a wound, the lesion itself is green.
Literally green. The tissue, the exudate, the pus, it has a distinct green color. Now, this is incredibly rare. There are only a handful of documented cases in the medical literature.
And it almost always happens when someone has a surgical wound or a major traumatic injury, and it gets exposed to contaminated river water or lake water containing these algae.
Now, as a clinician, if I see green pus in a wound, my brain immediately screams Pseudomonas aeruginosa. Pseudomonas bacteria are famous for making that pyocyanin pigment, which is blue-green. So how do you know you're dealing with chlorella and not just a very common Pseudomonas infection?
Well, the smell is a good first clue. Pseudomonas has a very classic odor. Some say it smells like sweet grapes. Others say corn tortillas.
Yes, the tortilla smell. But microscopically, the difference is obvious and immediate. Pseudomonas are these tiny, tiny gram-negative rods. Chlorella, on the other hand, are large eukaryotic cells. They're about maybe 4 to 10 microns across.
And when you look inside those large cells under the microscope, you can actually see these refractile green granules. Those are the actual chloroplast packed with starch. Okay, so green pus plus water exposure equals chlorella. Now let's talk about the cousin that lost its way, protozoa.
Protozoa is essentially a chlorella alga that, somewhere along its evolutionary path, lost its chloroplasts. It became what we call a chlorophyllus.
Because it can't make its own food via photosynthesis from the sun anymore, it had to adapt to survive.
Exactly. It became a saprophyte, living off decaying organic matter in the soil and water. And occasionally it becomes an opportunistic parasite of humans and animals.
And protozoa is much more common than chlorella. If you see an algae question on your board exams, there's a very specific high-yield clinical vignette you need to look out for.
Yes, the gardener's elbow. This is the absolute classic presentation for protophycosis. You have a patient, usually immunocompetent, often an older individual who loves gardening or landscaping.
They're constantly resting their elbows on the damp soil, or maybe they get a minor scraping injury on their elbow from a rose bush.
And weeks later, they develop a chronic olecranon bursitis.
So that's a boggy, swollen, fluid-filled, tender elbow joint.
Right. And they go to their doctor, who assumes it's a standard staph or strep infection, so they get prescribed standard antibiotics. But it doesn't heal. The swelling persists.
So eventually, orthopedics goes in and drains it. And the fluid they pull out is often serous sanguineous, kind of a watery, bloody mix. And when the lab cultures that fluid, they get these creamy white colonies growing on the plate that look exactly like yeast.
But they aren't yeast, they're algae.
So how do we spot them under the microscope to prove it's prototheca?
You look for the marula form.
The marula. Explain that visual for the listener.
Prototheca reproduces in a very unique way called internal septation. The mother cell doesn't bud. Instead, it divides its internal cytoplasm into multiple wedge-shaped endospores.
It looks exactly like the geometric panels of a soccer ball. Or if you imagine slicing an orange in half and seeing all the segments packed tightly inside the skin.
Some pathologists also describe it as looking like a daisy flower.
So if you see a description of a soccer ball cell or a daisy-like sporangium in the joint fluid from a gardener's elbow, you are dealing with prototheca.
Precisely. And the treatment is notoriously tricky. Because it's an alga, it's resistant to a lot of our standard antimicrobial drugs. We often try to use amphotericin B or even antibacterials like tetracyclines, but medical therapy frequently fails.
They have to cut it out.
Yeah, a surgical birsectomy physically removing the infected bursa is often the only way to actually cure it.
Okay, so to recap the algae segment for you. If it's green and from a river injury, it's chlorella. If it's colorless soccer balls from a gardener's swollen elbow, it's prototheca.
You got it perfectly.
Let's stay with the aquatic theme, but we're going to travel down to the Amazon basin. Segment 4, the Dawson disease. Laccatiosis.
Formerly known as lobomycosis. It was named after Jorge Lobo, the Brazilian dermatologist who first discovered it in the Amazon rainforest back in the 1930s. The organism responsible is Laccatia loboi.
And this pathogen connects humans and dolphins, which is just such a remarkably strange zoonotic link.
It really is. It naturally infects both the Atlantic bottlenose dolphin and the Guyana dolphin, causing these massive skin lesions. And in humans, it is strictly a tropical disease. You only see it in the Amazon basin and parts of Central and South America.
The clinical presentation in humans here is very distinct because it's not an ulcer. It's not a weeping abscess.
Oh, it presents as a keloid, or at least a lesion that perfectly mimics a keloid scar. The infection usually starts as a small, hard, painless nodule on the skin, typically on the legs or the arms or the ears, areas that are exposed to water and jungle vegetation.
And it grows, but it grows painfully slowly.
Yes, we are talking about a disease course spanning 40 or 50 years.
Half a century of continuous infection.
Yes. Over those decades, these initial nodules slowly merge together. They form these shiny, hard, raised plaques that look exactly like hypertrophic keloid scar tissue. They usually don't hurt, but they can be incredibly disfiguring for the patient.
Now, biologically speaking, Laccatia loboi is a yeast, but it has a very unique way of budding.
Right. Standard yeasts like Candida or Cryptococcus, they bud, and then the daughter cell pinches off and floats away to start its own life. Laccatia buds, but the baby yeast cell stays attached to the mother, and then that baby buds and its baby stays attached.
So you end up with a long chain of cells.
A chain of slightly elongated, lemon-shaped cells, and they are all connected by these tiny little narrow tubular bridges. The visual anchor you need to memorize for the boards is the string of pop beads.
Oh, like those plastic beads kids play with that pop into each other.
Exactly like that. Or you'll hear it described as a chain of lemons.
And the specific morphology is crucial for differentiating it from another major South American fungal infection, paracoccidioids.
Yes, paracoccidioidomycosis. Paracocci is also found in South America. It also causes chronic skin lesions, but paracocci has the classic pilot's wheel appearance under the microscope. It's a large mother cell with multiple little buds all around the outer rim, like a ship's steering wheel.
Whereas Laccatia is strictly a linear chain.
Exactly. A linear chain of lemons.
So how do we treat these massive keloids?
It's incredibly frustrating. Laccatia loboi has never been successfully cultured in vitro. No laboratory has ever been able to grow it on an agar plate. We only know it exists from examining infected human and dolphin tissue.
And because we can't grow it, we can't easily run drug susceptibility panels on it.
Right. Standard systemic antifungals like itraconazole have very limited, very mixed success. The only real definitive cure is wide surgical excision, physically cutting those thick keloids completely out of the skin.
But since the lesions are keloid-like and the margins can be fuzzy, I assume the recurrence rate is high.
Very high. It's a stubborn, lifelong companion for many agricultural workers and river dwellers in the tropics.
Okay, moving from the stubborn but slow to the aggressive and deadly. Segment 5. We are wading deep into the swamp now to talk about the water molds, Pythium and Legionnairium.
Listen, if I had to pick one organism from today's entire list of misfits that genuinely keeps me up at night, it is Pythium insidiosum.
Really? More than the AIDS pneumonia we talked about earlier.
Absolutely, because Pythium strikes completely healthy, immunocompetent people, and it moves incredibly fast.
Let's clarify the taxonomy first, because this is the ultimate wolf in sheep's clothing for pathologists.
It is an oomycete. It belongs to the kingdom Stromanopola. Genetically and evolutionarily, its closest relatives are actually brown algae and diatoms. It is not a true fungus.
But it desperately wants to look like one.
It does. Under the microscope, in a tissue biopsy, it produces hyphae. They are broad, ribbon-like hyphae, and they have very few cross-walls or septations. To a pathologist looking at a slide, this looks and I mean indistinguishably like the zygomycetes, things like mucor or rhizopus.
And that mistaken microscopic identity is often fatal for the patient.
It is, because oomycetes, much like Pneumocystis, do not have ergosterol in their cell membrane. If the pathologist misdiagnoses this as a standard mucormycosis infection, and the clinical team immediately starts high-dose IV amphotericin B,
which is the gold standard for mucor.
right, you are pouring highly toxic, kidney-damaging drugs into a patient, and it will have absolutely zero effect on the Pythium infection.
And meanwhile, the Pythium is spreading. And it has a terrifying predilection for certain types of tissues.
It is highly angioinvasive. The organism lives in swampy water and flooded grasses. It produces motile zoospores.
Wait, motile, like swimming spores?
Yes. They have dual flagella. They literally swim through the water. They seek out human or animal skin, usually entering through a minor cut or scrape on the leg. And once they get inside the tissue, they actively seek out the arteries.
They hunt the blood vessels?
They penetrate right through the arterial wall. And the human body responds to this massive structural breach by triggering a severe thrombotic cascade. The infected artery completely clots off.
Which means all the tissue downstream of that clot suddenly loses its oxygen and blood supply.
Exactly. You get rapid onset ischemia, wet gangrene, mycotic aneurysms, and massive necrosis of the affected limb.
So the clinical picture here is a young, otherwise completely healthy patient, maybe a rice farmer in Thailand, or a teenager who went swimming in a stagnant lake in Texas or Florida, who comes into the ER with a leg ulcer that is rapidly turning black, and the necrosis is spreading up the leg by the hour.
And because the antifungals they are given don't work, the only way to save their life is often immediate radical amputation. You have to surgically cut high above the visible line of infection to make sure you get all the angioinvasive hyphae. The overall mortality rate is distressingly high.
Is there any medical treatment at all that works?
Since our standard antifungals don't work, we have to get creative. There has been some clinical success using a combination of tetracycline and minocycline.
Antibiotics again.
Yes, those specific antibacterials seem to have some anti-oomycete activity. And interestingly, because this disease is actually quite common in horses, they call it swamp cancer. Veterinary medicine developed an immunotherapy vaccine for them.
Oh, wow. A vaccine to treat an active infection.
Yes, it ramps up the host's cellular immune response to recognize the oomycete proteins. And that equine vaccine has actually been used off-label in human patients on a compassionate use basis with some anecdotal success. But really, early and aggressive surgery is the cornerstone of survival.
That is heavy. So for your exams, Pythium equals oomycete, swimming spores, angioinvasion mimicking mucor, and ergosterol negative.
Correct. And you should also briefly know that Legionnairium is its close cousin. Very similar biology. It's also an oomycete, also causes severe angioinvasive disease. It's a bit more common as a pathogen in dogs, particularly hunting retrievers plunging into swamps. But it can and does infect humans, too.
All right, let's pull ourselves out of the swamp and dry off for our final misfit of the day. Segment 6, rhinosporidiosis.
This one easily wins the award for the most visually striking and honestly arguably the most grotesque clinical presentation.
The strawberry nose.
Yes. The organism is Rhinosporidium seabury. Once again, despite being in the mycology textbook, it is not a fungus. It belongs to a very obscure taxonomic group called the Mesomycetozoa.
Which is a fantastic name. It literally translates to between fungi and animals.
It's an aquatic protist. Usually, organisms in this class are parasites of fish and amphibians.
But in humans, particularly in endemic regions like India and Sri Lanka, it causes these massive fleshy polyps.
It's primarily a mucosal infection. The nasal cavity is by far the most common site, followed by the conjunctiva of the eye. A patient will present with a large, friable, highly vascular red mass physically hanging out of their nostril.
And friable means it bleeds very easily if you bump it.
Right. It's very fragile tissue.
And if you look closely at the surface of this red polyp, you see these little tiny white dots speckling the surface.
That is the classic tapioca sign. The combination of the red vascular tissue and the white dots makes the polyp look exactly like the surface of a strawberry or a scoop of tapioca pudding.
What exactly are those white dots?
Those dots are the mature sporangia of the organism. And this brings us to the microscopic heavyweight champion of the episode. Earlier, we talked about mycology being huge at 400 microns. Rhinosporidium sporangia are nearly that big. They grow up to 350 microns in diameter.
So again, you can easily see the individual sporangia with the naked eye.
But unlike the empty balloon of mycology we talked about earlier, these sporangia are packed to the brim. They are bursting with life. A single mature sporangium contains thousands upon thousands of tiny endospores. And what's unique is they have a very specific zonal arrangement.
Explain that zoning for us. How does it look on a slide?
Inside this giant spherical sac, the endospores actually mature at different rates depending on their physical location. Usually you have the smaller, immature endospores packed tightly around the outer periphery of the sac, and the larger, fully mature endospores are clustered in the center.
Or sometimes vice versa, depending on the plane of the slice.
Right. But it creates this very distinct visual layering or a zonal effect inside the cyst when you look at it under the microscope.
So let's differentiate this for the listener, because this is a classic board question setup. You're looking at a biopsy slide, and you see a large sphere full of endospores. Is it coccidioids, or is it rhinosporidium?
You have to look at two things: location and size. Coccidioids is going to be found in the lung tissue, and its spheral is smaller, maybe 60 to 100 microns max. Rhinosporidium is going to be found in mucosal tissue, like the nose or the eye, and the sporangium is absolutely massive, up to 350 microns.
Also, rhinosporidium cannot be cultured on standard media, whereas coccidioids grows very easily and very dangerously for the lab techs on standard agar plates. And what is the treatment for the strawberry nose polyp?
Unsurprisingly for this chapter, the primary treatment is surgery. You have to physically excise the entire polyp. And crucially, the surgeon has to deeply cauterize the base of the lesion because local recurrence is extremely common if any tissue is left behind.
And I'm guessing systemic antifungals are useless here too.
Useless?
Because it's a protist, not a fungus.
You are definitely sensing the major theme of Chapter 66 here.
I certainly am. I mean, we have covered a tremendous amount of weird rule-breaking biology today. We've gone from protozoa that are actually fungi without ergosterol, to algae infecting gardener's elbows, to swimming water mold spores destroying arteries.
It really highlights the fact that nature does not care at all about our neat little textbook chapters.
It absolutely doesn't. Now, before we completely wrap up this deep dive, we need to solidify this knowledge. It is time for the rapid-fire single best answer quiz.
The consolidation phase?
Exactly. I'm going to read short clinical vignette. I want you to give you the diagnosis and, most importantly, explain the key hook or mechanism that gives it away.
I'm ready. Let's do it.
And for you listening, pause the audio briefly after the question if you need a second to think before we reveal the answer.
Oh, Eureka.
Question 1. A 35-year-old male with untreated HIV and a CD4 count of 80 presents with severe shortness of breath and a dry, non-productive cough. He is severely hypoxic, but his lungs sound mostly clear on auscultation.
Yeah.
A silver stain of his induced sputum reveals cup-shaped cysts resembling crushed ping-pong balls. He's admitted and started on high-dose IV fluconazole, but he shows absolutely no clinical improvement after 4 days. Why did the treatment fail?
All right, the diagnosis here is Pneumocystis pneumonia, or PCP. The treatment failed because Pneumocystis jirovecii completely lacks ergosterol in its cell membrane. Fluconazole works by targeting ergosterol synthesis. Since this organism scavenges host cholesterol instead, the antifungal drug has no biochemical target. The patient needs trimethoprim sulfamethoxazole to target its folate pathway.
Perfect. Question 2. A 60-year-old avid gardener presents with a chronic, swollen, very tender lesion on his right elbow. He remembers grazing the elbow against a dirty rose trellis a few months ago. The fluid is drained, and a wet mount biopsy reveals eukaryotic spherical structures with internal wedge-shaped divisions that closely resemble a soccer ball. What is the organism?
This is protothecosis, caused by Prototheca wickerhamii. The key clinical clues are the gardener history, the location on the elbow indicating olecranon bursitis, and the microscopic description of the morula, or soccer ball morphology. Remember, this is in a chlorophyllous alga, not a yeast.
Excellent. Question 3. A marine biologist who has spent decades working in the Amazon basin presents with a series of hard, raised, shiny nodular lesions running up his forearm. They are painless, they look exactly like keloids, and he states they have been slowly growing for the past 10 years. A tissue biopsy shows chains of yeast-like cells connected by narrow tubes. What is the diagnosis?
This is leukocytosis, also known as lobomycosis. The triad here is the Amazon rainforest or dolphin exposure history, the slow-growing keloid-like lesions, and the microscopic finding of chains of lemons, or the pop beads appearance. That is diagnostic for Leukocia loboi.
Spot on. Question 4. A healthy young man in Florida cuts his lower leg while swimming in a stagnant grassy pond. Within days, the wound becomes necrotic and a black discoloration spreads rapidly up his calf. A biopsy shows broad, ribbon-like aseptate hyphae invading the blood vessels. He is empirically treated for mucormycosis with amphotericin B, but he continues to deteriorate with spreading arterial thrombosis. What is the actual pathogen?
The organism is Pythium insidiosum. The setup of mucor-like broad hyphae combined with the absolute failure of amphotericin B, plus the aquatic swamp exposure and the aggressive angioinvasion, equals pythiosis. It is a deadly umicete, and he likely needs an immediate amputation to survive.
Question 5. A patient from a rural village in India presents with a large, friable, bleeding red polyp hanging from his right nostril. He mentions he bathes daily in the local stagnant village pond. Pathology of the excised mass shows a massive 300-micron sporangium containing thousands of endospores with a distinct zonal arrangement. What is the disease?
This is rhinosporidiosis, the presentation of a strawberry-nosed mucosal polyp, the stagnant water exposure in India, and the microscopically massive sporangia, much larger than coccidioids, is the key diagnostic triad.
Question 6. An asymptomatic patient has a solitary coin-like lung nodule removed after it was spotted on a routine chest X-ray. Pathology reveals a single, massive, 400-micron thick-walled sphere embedded in a granuloma. Crucially, this sphere is completely empty on the inside. What is the organism?
This is adiasporomycosis, caused by Adiasporomyces crescens. The visual of the balloon that never pops is your anchor here. It's defined by its huge size, its incredibly thick wall, and the fact that it contains no endospores inside.
And finally, question 7. A patient presents with an infected surgical wound on their leg after falling into a river during a rafting trip. The pus draining from the wound is bright green.
This is chlorellosis. The bright green color is the giveaway. It comes from the active chloroplasts inside the chlorella algae that are infecting the wound tissue.
Outstanding. I hope you listening nailed those along with us.
And if you missed any of those hooks, please go back and review the specific sections in Murray chapter 66. These are incredibly high-yield associations because they are so biologically unique. They make for perfect exam questions.
So we have officially survived the island of misfit toys.
We have. And you know, what I find most provocative about this entire chapter is what it actually says about our own scientific ignorance.
How so? What do you mean?
Well, think about it. We spent decades, literally generations of medical training, confidently calling Pneumocystis a protozoan parasite. We called Pythium a standard fungus. We treat these taxonomic boxes as if they are absolute truths. But we are constantly having to reshuffle these boxes every time new DNA evidence or molecular sequencing emerges.
That's a really good point. The organisms haven't changed in millions of years, but our understanding of them just flips overnight.
Exactly. It really makes you wonder, what common fungus are we treating in the hospital today that is actually something else entirely? What organism is sitting right now in chapter 40 of our textbooks, totally misclassified, just waiting for a new generation of DNA sequencers to reveal its true identity?
That is definitely a thought to sleep on. Or maybe not sleep on, considering some of these bugs.
Indeed. Biology is always weirder than we think it is.
It certainly is. As always, for the listener, we highly encourage you to open the textbook alongside this audio. You really need to see the images of the Prototheca morula, the Leukocia chain of lemons, and the crushed ping-pong balls of Pneumocystis to truly lock them into your visual memory.
Visual anchors are everything in microbiology.
They really are. Well, that wraps up our coverage of mycology's strangest outlier.
Until next time.
Keep digging deep.